Hello everyone, my name is Charlotte. I am a first-year PhD student in SITraN at the University of Sheffield. I am currently researching the most common genetic cause of motor neuron disease, which is a mutation in a gene called C9orf72. This mutation is a repeating sequence which produces proteins. These then accumulate in the central nervous system and are thought to cause toxicity. This mutation can be repeated many times. In healthy people, it can be repeated up to about 30 times, but in people with motor neuron disease, it can be repeated hundreds or even 1,000 times. Our lab uses the first animal model to express the mutation at this disease-relevant length. Our model is a fruit fly. While a fruit fly may not seem very similar to a human on the surface, there are actually many similarities, from the structure to the nervous system down to genetics. Flies have been used as a model in medical research for many years, including for cancer research, and as a common model in neurological research. When we zoom in on cells, many functions and structures are found in both flies and humans. One structure is called the cytoskeleton, which can be seen in pink in cells here. This gives cells their shape and is required for transport of important materials around the cell. In motor neuron disease, we can see that this structure becomes disorganized. This leads to cell death, which in our flies causes movement impairment and a reduced lifespan. When we treat our flies with cytoskeleton stabilizing drugs, these impairments are reduced as well as the cell death. The aims of my project are therefore twofold. First, I am aiming to understand the mechanisms behind disease, how these proteins are disrupting the cell structure, and why this is causing disease symptoms. Second, I am investigating the possibility of treatment, either genetically or by repurposing clinically approved drugs to stabilize the cytoskeleton. This, along with the mechanistic understanding, may reveal potential drug targets and open avenues for therapy. Thank you to my supervisors, my lab, my funders, and thank you for listening.